I am very glad to be giving this talk today. As a caveat, I am a breast medical oncologist, so I primarily clinically focus on breast cancer. However, I direct our cancer center's Johns Hopkins Molecular Tumor Board. And so in that setting, we review cases from across the cancer center for all tumor types and review the next generation sequencing or molecular profiling from all solid tumors. So I'm going to be focusing on discussing somatic versus germline, which is a very important distinction to be making when we're thinking about inherited versus acquired mutations in melanoma. Then discussing some of the germline susceptibility genes and syndromes in skin cancer, but with a focus on melanoma, and then covering a little bit about inherited susceptibility in melanoma and who to test for these germline susceptibility genes. And then discussing some of the somatic mutations that we can see in melanoma, and then followed by take-homes. So this is a really important distinction to understand when we're talking about genetic changes in any cancer type, including melanoma. And this is the germline versus somatic. On the right, we see what are germline DNA changes. What that means is that these are DNA changes that were present at birth, meaning they're present in every cell in the body, including the egg and the sperm, and were passed on from a parent and can be then passed on to children. They increase cancer susceptibility, so they're inherited mutations. And then in contrast, there are somatic DNA changes which were not present at birth and are acquired in a person's lifetime in single cells in the cancer. They can lead to cancer and they cannot be inherited or passed on. So what are some common differences between germline and somatic? I kind of made this table here to make the distinction. So in germline mutations, this is what it asks is, was I born with a gene change that increases my cancer risk? Whereas with somatic means what gene changes are in my cancer right now. The testing is different between the two. So to test for germline mutations, that's typically on a saliva or sometimes on a blood sample, whereas somatic mutation testing occurs on the tumor tissue itself. And now more and more through blood, what's called a liquid biopsy, which is looking at tumor DNA that's shed into the bloodstream that can sometimes be picked up on a blood sample. Germline, the purpose is to find an inherited risk susceptibility, and it can guide further screening for the melanoma or other cancer types, whereas somatic guides treatment choices and clinical trials. Germline, again, may apply to relatives, where somatic usually does not apply to relatives. And with germline testing, that's usually done once, sometime at diagnosis, whereas somatic can be repeated multiple times as the DNA and RNA changes evolve in the course of a patient's cancer treatment. So this is a big table of germline predisposition genes and skin cancer. And we're not going to go over all of this today. Each one of these could be a separate 10-minute talk. But these are some of the syndromes or conditions and their associated genes that are associated with skin cancer. We're going to be focusing on melanoma primarily. As Dr. Topalian mentioned, most melanomas are not inherited. In fact, only 8 to 12% of patients with melanoma have a family history of the disease, but not all of these patients have hereditary melanoma. So for example, you can be part of a family that has a common behavior or a common heavy sun exposure and a susceptible skin type. So it's not that they have an inherited gene, but rather something else that's making the family more susceptible to melanoma. Within inherited susceptibility genes, there are multiple, and we won't go over all of them, but they're classified into what's called high penetrance versus intermediate penetrance. high penetrance means that if somebody has this inherited gene, then it's likely that they will develop melanoma at some point in their lifetime. Whereas intermediate or low penetrance genes are an inherited gene, but that doesn't increase the likelihood of getting melanoma by much. One of the most common inherited susceptibility genes is this mutation in what's called CDKN2A, which is mutated in around 20 to 40% of high-risk families. CDKN2A, which is shown at the top of this figure here, is a gene that encodes two proteins, P16-INK4A and P14-ARF. And each of these proteins is responsible for regulation of the cell cycle. So P16-INK4A, when it is mutated, it causes uncontrolled growth and proliferation through the cell cycle. So basically, it allows the cells to divide unchecked. Whereas a P14-ARF controls P53, which is involved in apoptosis or cell death. And so when that's mutated, then the cells don't die. So essentially having a mutation in CDKN4A leads to a dysfunctional protein, which allows the cells to divide and grow abnormally. The penetrance, for individuals who carry a germline CDKN2A mutation varies from, in some studies, as low as 28% to more than 90% by age 80 years of age. So basically, if you have this mutation, it means by age 80, up to 90% of those individuals will have an experience of melanoma. So who to test for hereditary melanoma? Obviously, speak to your physician, dermatologist, or medical oncologist about this, but in general, there are some guidelines about who to test because, again, the majority of people will not have an inherited gene that predisposes them to melanoma, but there's different criteria depending on whether that person lives in a geographic area with low melanoma incidence or moderate to high melanoma incidence. In the US, we're considered a high melanoma incidence geographic region. Other areas like Australia are also considered that. But in high melanoma incidence, basically, if you have had melanoma 3 times, 3 primary melanomas, or families with certain clinical features in first or second degree relatives. So family history of melanoma, or pancreatic cancer, because that's related to the type of the CDKN2A can also increase the risk of pancreas cancer. But in general, the bottom here I've summarized who to consider for testing. These are people who have been diagnosed with melanoma at an earlier age, have relatives, multiple relatives with melanoma. You yourself have a melanoma, multiple primary melanomas, or certain other cancer types in your family. Now we're going to move on to somatic mutations. So again, these are acquired mutations that happened in the cancer, that are happening in the cancer cells themselves. And again, a long list of potential somatic or acquired mutations in melanoma. We won't be reviewing all of them, but we'll discuss in the next few slides some of the more common somatic mutations that we see. The most common is BRAF mutations. BRAF, which is shown in the right there in the blue in the middle, is a type of kinase in what's called the MAP kinase signaling pathway. It regulates cell growth, survival, and differentiation. And basically, mutations in BRAF lead to abnormal pathway activation, which causes more cell growth and survival. This occurs in about 40 to 50% of cutaneous melanomas, this BRAF mutation. And the most common mutation is a mutation called BRAF V600E. This is clinically relevant because there are FDA-approved therapies that target BRAF mutations. including the ones that I've listed below. These are called BRAF-MET combination therapies. KIT mutations are also a relatively common somatic mutation that occurs in melanoma. KIT, which is that purple Y-shaped structure up there, is a gene that makes a cell surface protein that acts kind of like a growth switch. So when it's mutated, the growth switch turns on and leads to more cell growth and activity. This mutation occurs in about 1 to 3% of all melanomas, but is enriched in certain types of melanoma, such as acromelanoma and mucosal melanoma. Also clinically relevant because there has been some efficacy of a targeted therapy called imatinib or Gleevec against mutations in KIT. And another common mutation is NRAS, which is another receptor tyrosine kinase that is above BRAF. These also activate downstream signaling pathways in MAP kinase and PI3 kinase that lead to abnormal cell cycle dysregulation and dysfunction and abnormal growth and proliferation. This mutation occurs in about 20% of cutaneous melanoma. At the moment, people have tried to target NRAS, but it's been challenging. Dr. Warrior will be talking more about treatment in melanoma and clinical trials, but this is another common mutation that's seen in melanoma. So take-home messages, again, though most skin cancers are not related to an inherited susceptibility gene, you should discuss with your care team that two primary questions: should I or my family be tested for germline or inherited mutations? And if one does have a diagnosis of melanoma, most commonly this is important in metastatic melanoma. Is tumor next generation sequencing appropriate? And how will those results change my treatment or my screening?